Okay, welcome back. This is part 3 of today's screencast lecture, where we'll move on and wrap up the material for Tuesday. So we're kind of back to square one now. These endogenous rhythms are part of the solution, but they're not accurate on their own. So now we're back to looking for environmental cues to work with the endogenous rhythms to give us the precision that we need. What we need is an environmental cue that changes precisely and predictably every year. It can't be something like temperature that fluctuates. And there's an ideal candidate out there. Again, here's a graph showing an annual cycle with a variety of events up above, and then down below is that environmental variable that changes by a predictable amount every day and is consistent from year to year. That is day length. And that's our best option, it seems, right now for that environmental cue that's going to help us determine what the time of the year is. So, minor problem. Day length is an ideal environmental variable for determining when in the year we are, but how do you measure day length? Just like birds don't carry calendars, they don't carry watches either. The key here is going to go back again to endogenous rhythms that exist. And in this case, endogenous rhythms that are not on a yearly cycle, but on a daily cycle. These are referred to as so-called circadian rhythms, or circadian. So in the same way our circannual were about a year, these are circadian, or about a day. The figure here is showing activity for a bird called a chaffinch, again a European species, with at the beginning here activity patterns on a normal light-dark cycle that the bird's kept in captivity. The lights go on, it becomes more active. The lights go off, and it becomes less active, more active, and so forth. Nice cycle going on there. And then the second part is the birds are then put under a constant lights-on condition, where the environment stays the same, but you can see the same fluctuations start to happen of high activity for about 12 hours followed by low activity for about 12 hours in the absence of that light-dark cue. These are circadian because, again, it's not exactly 24 hours, and in fact, typically what we're looking at are about 23-hour cycles. So we've partly started to solve our problem, but now we also need to make the clock more precise and entrain it. And we do this with something called Zeitgebers, which, again, one of those German terms. In this case, Zeit, time, Geber, giver. So it's a cue that gives the time. So how does this work? The short answer is we're not entirely certain of all the details. Gill provides probably the best hypothesis that I know of in your reading for today on pages 253 to 255. Where this starts is with a circadian rhythm, that is an endogenous rhythm about a day, and in particular a circadian rhythm in photosensitivity, or how sensitive the bird is to daylight. The model that emerges from this, Gill shows in figure 9-9, and I'm going to go through it here. It's a little complicated, but it's something that I want you to know for the exam. So the light-dark bars here represent different light-dark cycles at different times of the year. So at the top here might be the situation during the winter, when there's only 8 hours of daylight and 16 hours of dark. Get into spring, when you've got 16 hours of light, 8 hours of dark, and this bird's really far north, where we might have 22 hours of daylight at mid-June, for example. So that's these light-dark bars going across. the circadian rhythm and sensitivity is shown by these curves, where in the winter, the bird's most sensitive to light in the time when it should be dark, that is at night. And that's consistent in all three cases here. What changes is as day length gets longer, the amount of time that the bird is exposed to light when it's sensitive to light increases. And that could then trigger a response. Okay? That's the general idea of this hypothesis. And again, it's something I want you to be able to explain. The next step we need for this to work, however, is something to measure light intensity. Now, surprisingly, the primary organ is not the eyes, but actually the hypothalamus that's in the center of the brain, along with some input from the pineal gland in the brain, and to a lesser extent, light receptors in the retina. Now, the fact that these are the light receptors is actually pretty well supported. Variety of levels of or types of evidence. This being a diagram here, a cross section of the brain, the cerebrum, the cerebellum, and then here in the middle of the brain is that hypothalamus. And we know this is an important light receptor that controls these daily and annual cycles because, for example, if you do surgery on a bird, you remove the hypothalamus, it loses the cycles. On the other hand, if you take a very fine optical fiber and just pinpoint light only at that hypothalamus, you can induce things like breeding behavior and migratory behavior, et cetera, et cetera. Now, for the next step in this process, it helps to remember that the hypothalamus and the pineal are part of the endocrine system. That is, the system that controls the body's activities using hormones. And in fact, stimulation of these glands by light causes the release of hormones that can influence what birds do. The specifics of what goes on next depend on what activity in the annual cycle we're interested in. As often happens in the case of the endocrine system, controls are driven by a cascade of events with one hormone acting on another part of the body to produce another hormone, so on and so on. A typical sequence event for something like reproduction occurs when this hypothalamus is exposed to light. Under our model that we went through earlier, it's exposed to light during the photosensitive period of the daily cycle. And when it's exposed to light, it releases a hormone known as LHRH, the luteinizing hormone release hormone. That's a mouthful. And this hormone, released by the hypothalamus, actually acts on an adjacent part of the brain, the pituitary. So it doesn't travel very far, but it induces the pituitary to in turn release two hormones, often called master hormones. Here, Gill uses this terminology. And in particular, it reduces LH, luteinizing hormone, and FSH, follicle stimulating hormone. And these travel through the body and in turn act on different parts of the body. In males, luteinizing hormone stimulates the Leydig cells to produce testosterone. Testosterone then leads to enlargement of the testes and puts that male into breeding condition. In the female, luteinizing hormone induces enlargement of the ovary. Follicle stimulating hormone acts to stimulate sperm production and egg production in males and females, respectively. So again, we've got a cascading series of events here for, in this case, reproduction. Table 9-1 in your reading by Gill lists other hormones that control other activities. And as one example, remember that part of this annual cycle is keeping activities separate. And there's a good example here where we've got a hormone produced by the thyroid, thyroxine, that is responsible for causing molt. However, this only happens if testosterone levels are not too high. So during the breeding season, when testosterone is being produced as a result of this cascade of luteinizing hormone release hormone, luteinizing hormone producing testosterone, it inhibits the production of the thyroxine so the birds don't molt while they're breeding. Once breeding season is over, those testosterone levels decline, thyroxine is produced. Now we start having molt going on. Now this process is actually quite sensitive and quite precise. We can see this by looking at some birds and how they respond under different conditions. A great example are those birds that live fairly close to the equator, where there isn't a dramatic change in day length. This is a spotted antbird that lives in Panama, just north of the equator. And you can see from this graph that the difference between the shortest day of the year and the longest day of the year is only about an hour in length. And yet the top graph shows this same cycle of testis size and other variables as well. So the bird's going through the cycle and yet is not exposed to dramatic changes in day length. And in this study, they brought the birds into captivity and experimentally exposed them to different light regimes. And again, we'll just look at testis volume in this case. The open bars are birds exposed to a 12-hour light, 12-hour dark daylight. And then when birds were exposed to 13 hours light and 11 hours dark, they started getting into breeding condition. And in fact, this happened at a much shorter difference. If we extended the amount of light from 12 hours to 12 hours, 17 minutes, the birds showed this response of going into breeding condition. So very precise. We might not know all of the details of the model, but it's well supported that it's there and functioning in the way that we expect it to. All right, that's it for today in this lecture. Reminder, what I'm going to want you to know is the typical annual cycle of birds, why birds do things when they do at specific times of the year, that is, that variable environment, the need to time important events to occur at the most favorable time of the year, that need to keep events separate from one another. So you'll be seeing things like the graph of the birds in Borneo, the graph with the 5 species in relation to the food supply. Those will be on your handout that you can download. And then I want you to know about how all of this is controlled. That calendar and clock, that we've got these circannual events, but they drift and need to be entrained. We've got circadian, or about a day, cycles that also drift and need to be entrained by the Zeitgebers, the time givers. That model of how changes in photosensitivity on a circadian basis can then lead to the control of these events through the cascading effects of hormones released by the hypothalamus impacting the pineal and so on. So be sure and download the handout. Again, much of this we'll come back to when we talk about specific events in the annual cycle. Migration is next up. We'll talk about reproduction later, and so you'll see a lot of this material again. If you have questions, talk to me on Thursday. Otherwise, stay warm, and we'll see you at lab at 7:30 on Thursday morning.